Hello friends, in this video we're gonna study about kingdom fungi. So kingdom fungi is one of the kingdom according to Whitaker's classification. So Whitaker has classified into 5 kingdoms, right? And the first is Monera, Protista, then kingdom fungi, Plantae and Melia. So one of these is kingdom fungi and we are going to study about it. So I am covering here the complete NCERT of class 11. So you don't have to worry about any competition exams. Your NEET syllabus will be completed completely here. Okay friends. So first of all, this kingdom fungi is also known as kingdom mycota. And the study of these fungi is called as mycology. Why it is called as mycology? So if you see this word mycology, it is having a prefix and a suffix. In prefix, this myco word has been taken from Greek language, which means mushroom or fungus. Simple. And logy. What does logy means? To study. So here we are going to study about fungi. That's why it is called as mycology. Okay. So let's study about some general features. Then we will go about the particular study of particular groups in the kingdom fungi. So first, the mode of nutrition, how they obtain food. So fungi, they don't have the chlorophyll pigment, so they cannot synthesize their own food. So they have to depend on other organisms for food. So that's why they are called as heterotrophic organisms, right? So in the heterotrophic organisms, there are different mode of nutrition. So let's see about the different mode of nutrition in the fungi. First of them is saprophytic mode. So what is saprophytic mode? You must have heard of it earlier in the bacteria also. So these are the organisms which grow on dead organic matter, or you can say like they grow on the dead bodies, like vegetable waste, fruits, like or other organisms body. They can even grow on the fallen leaves. So these are all dead organic matter formed of organic compounds. So these saprophytes grow on these dead organic matters and obtain their food. So how they obtain their food? They release the enzymes. So what they do? They release the enzymes and break down the complex food. So they are releasing enzymes outside their body. Okay, they are releasing their enzymes outside the body to break down that complex compound. Okay, that complex compound is then converted into a simple product, and that simple form of food is absorbed by the fungi. It's that simple, right? And in this process, many nutrients are also given back to the soil, right? Okay, so this is the simple way. You have seen many of the dead organic matters, garbages, there these fungi usually grow. Okay, next, another mode is the parasitic form. What are parasites? Parasites are the organisms which grow on other organisms on their surface or inside their body. So those growing on the surface are called as ectoparasite, and those growing inside are called as endoparasites, right? So, fungi also grow over their host, right? So how they obtain nutrition from their host? So they have a special structure called hostoria, right? These hostoria are formed from the fungal branches. So here you can see they have formed this hostoria, and this hostoria has penetrated into the plant cell. From there they have absorbed the nutrients. So ready-made nutrients they are getting from the plants, right? So like this they obtain their nutrition. So these are the common mode of nutrition found in the fungi, right? Next comes the symbionts. What are symbionts? So those organisms living symbiotically, showing symbiosis. Symbiotically means both are benefiting each other, like algae and fungi both live together as a symbiont and called as lichen. In this relationship, algae manufactures food for the fungi, while fungi provides shelter to the algae and helps in absorption of nutrients, right? So both are benefiting each other in this relationship, and this type of relationship is called as symbiosis, right? And the another relationship you can see between the higher plants roots and the fungi. So fungi grow in the roots of higher plants, and this association is called as mycorrhiza. This association called as mycorrhiza. How they are benefiting each other? So fungi is absorbing nutrients for the plants like phosphorus and competing with the harmful pathogenic microorganisms, right? And what benefit fungi is getting? It is getting ready-made food from the plant, right? So that's why both are benefiting each other. Likewise, if you see the structure of the fungi, so you will find a long diversity. There are many different forms of fungi available. So here is the word written morphology. What that mean? Morphology. This is the external appearance of the organism, even an organ, like. And here we are talking about the fungi. So fungi are having different forms. Some you will find a cellular forms like yeast, which is unicellular, right? And some can be like filamentous form. And you have seen the structure of mushroom. It's like an umbrella, right? So likewise they have different morphology and they are found in different habitats too. So they are cosmopolitan. Cosmopolitan means almost everywhere they are available. So in air, water, soil, on animals and plants, parasitic forms are present on animals and plants, right? So their spores are spread everywhere, even around you these are present, right? But you can't see them. So they normally grow in the warm and humid places. Okay, where they are growing? In the warm and humid places. Okay, so these unicellular fungi, yeast. What is the scientific name of yeast? Saccharomyces, right? So this yeast, you must be knowing that they are used for the manufacturing of the bread making process and in the beer. So because we are using it in bakery set, that's why it is called as baker's yeast. And for the beer production it is also used. That's why it is called as brewer's yeast. Right? Okay, we will talk about it later on again. Next other form of fungi is the filamentous form. In this filamentous form, you have to remember some points. First thing, so this filamentous form has a long, slender thread-like structures. Here you can see this is the long thread, and this single thread is called as hyphae. Clear? Hyphae is this single thread, and when combining these all threads, a network is formed, and this network of threads is called as mycelium. Okay? Remember this thing. This network of threads is called as mycelium. Clear? So these hyphae can be aseptate or septate. What that means? Aseptate and septate. Aseptate means they are not having septa in between. Means septa means partition. But septate hyphae means there is a septa present in between. So here you can see in the septate hyphae there are septa present. Okay, that's why you can see these cells are uninucleated in the hyphae. But in cynocytic hyphae, what that cynocytic means, these are multinucleated because they don't have any septa. So there is no partition. If there is no partition, that means they are multinucleated. Many nuclei are present in a hyphae. Clear? So it's simple. Filamentous fungi can be septate or aseptate. We will find out which are septate and aseptate in the further study. Okay. Next is the cell wall. What is their cell wall made up of? So cell walls are made up of chitin. What is chitin? Chitin is a complex polysaccharide. This is formed of an acetyl glucosamine. Clear? Chitin is a polysaccharide formed of an acetyl glucosamine. Clear? And with it some polysaccharide molecules are also attached. So chitin itself is a complex polysaccharide. Clear? So Ncert writes it like it is composed of chitin and polysaccharide. What are polysaccharides? These are the complex carbohydrates having monomer units above 10. Right? Clear? Okay. Let's move to the next. So here we're gonna study about the reproduction in fungi. So how fungi reproduce? So it must be reproducing by different methods, like some vegetative or asexual method, some sexual methods. Like here the first one is the fragmentation. So fragmentation is a simple vegetative method in which a fungi hyphae break into pieces and each piece grows into a new hyphae and forms a mycelium. Clear? Simple. A fungal filament, or we can say hyphae, breaks into parts and each part grows into a separate fungi. Clear? So this is fragmentation. Examples are molds and mushroom. Clear? Next is the budding. Budding you have seen in the yeast. Okay, we have seen earlier picture. It is shown here. You can see that there many birds are arising. These are the birds which are formed. How these birds are formed? So these are the outgrowths. Clear? So it's a outgrowth on the surface and this outgrowth grows. Nucleus divides, one nucleus goes into the outgrowth, one remains in the parent cell, and this outgrowth grows into a separate individuals later on. Okay? So it's a simple process. A bud forms over a body and it grows into a new yeast cell. Simple. So it can be remain attached to the parent cell. In this case, they forms the chain of cells over the parent cell. Right? So it can be observed as a chain of cells. Clear? So budding is very simple. Right? A bud will be formed. So Saccharomyces yeast shows the budding. Next is the fission. Fission is the simple. There are two words, fission and fusion. Fusion means to combine, and fission means to break, separate apart, right? So here a unicellular form can break into two daughter cells. This is called as fission. Simple. A single cell is broken down into two daughter cells. It is called as fission, and this occurs in again yeast, right? Okay. There are different asexual modes in which spores are produced, and these spores germinate to produce a new organism or a new individual. So here, what is spore? Spore is a vegetative propagule, or we can say single-celled reproductive structures which on germination produces new organism. Okay? In the simple way I have defined it, you can remember it. Okay, so there are different spores. First is the conidia. So how conidia is formed and what are its like what is its structure and how it looks like. So first of all, you must know conidia are non-motile spores. Non-motile spores means they don't have motility, they cannot move their own. Okay, and they develop in chains. So here you can see like in Aspergillus and Penicillium, they are formed in the form of chain. So they are present in the form of chains. So they are formed in a specialized hyphae called as conidiophore. So they are developed on the conidiophores in the form of chains, right? Here you can see these are the conidia, and on germination they produce new individual, right? Simple. Sporangiospores. So sporangiospores, they are normally observed in the Rhizopus. You have seen a fungus growing on the bread, right? So that green colored fungus is Rhizopus. So these structures are showing formation of spores, and the specialized hyphae is called sporangiophore, on which a bulb-like structure is developed. this bulb-like structure is called as sporangium, in which sporangiospores are formed. And simply we can say like spores are formed which on germination produce new Rhizopus, right? So these spores are formed in sporangium. So you have to remember this sporangium. How are these spores? These are also non-motile spores. Why they are non-motile? Because they are released in the air and air currents will carry it. So they are light-weighted, very small, easily carried by air currents to the suitable surface where they can germinate again. Right? Simple. Okay. Likewise, there are other spores also like zoospores. So these are formed in the aquatic environment. These are the motile spores having flagella over their body. That flagella provide motility to them. So that flagella will move and the spore will start moving, right? So it's a very simple that zoospores are the motile spores having flagella and they are released in aquatic environment. Remember this thing that they are released in water bodies, okay? And specialized hyphae in which they are produced are called as zoosporangia, right? They are formed in the structure called zoosporangia. Example is Phytophthora. We will see the zoospore structure in the life cycle. We're going to study further. So in just 2 or 3 minutes it will come. In the life cycle also we will study about this. Next are the aplanospores. These are also non-motile spores having thin cell wall different from the parent cell. Okay, so these were the some of the spores I have discussed produced in the fungi. Okay, next is the sexual reproduction. How they are reproducing sexually. So there are different structures are formed during their life cycle, and first one of them is oospores. So what are oospores? They are a thick walled zygote. Okay, they are thick walled zygote formed from oosphere. Oosphere, what is oosphere? A female reproductive cell, or you simply say like na egg cell. So this is same oosphere. So this oosphere on contacting with male gamete means after fertilization they form zygote that is called as oospore. Okay, complete study we will do in the life cycle. So in the individual groups when we will study, there I will show you this also. Next is the ascospores. These are the non-motile spores produced in the asci. Clear? So here I have shown you ascus in which there are the spores. Okay, they can be from 4 to 8 in number. Okay, remember this thing. Now, the next is Basidiomycetes. So likewise, Basidiomycetes are also produced in sexual reproduction. So what are these Basidiomycetes? These are also non-motile spores produced exogenously. You have to remember this word exogenously. Exogenously means outward they are formed. Like here you can see these are formed outside the main body, but these ascus is produced inside the host, right? So these are produced inside the body, that's why they are called as endogenously produced inside the body. Exogenously means outside the body they are being produced. So you have to remember this thing. Clear? Next, what are the steps involved in sexual reproduction? So you have to remember these steps so that you can easily learn about their life cycle. So very first step is the fusion of the protoplast between two motile or non-motile gametes, and it is called as plasmogamy. So there are two hyphae, and these are haploid hyphae which fuse together, and this fusion is called as plasmogamy to form a dikaryon. Dikaryon is not present in every fungi. These are specially found in the Basidiomycetes and Ascomycetes. So you have to remember this thing that dikaryon stage, in which a cell is having two nuclei. Di means two, karyon means nucleus. So a cell is having two nuclei, that is called as dikaryon, and it is mainly present in Basidiomycetes and Ascomycetes. Clear? Now these two nuclei fuse together, and this fusion is called as karyogamy. Okay, this fusion is called as karyogamy. So by this a synkaryon is formed. Okay, having diploid chromosome. So this synkaryon will produce a fruiting body in which meiosis occurs to form haploid spores. So these are the haploid spores formed, and these on germination produce haploid hyphae. So it's clear that these are following a cycle. A haploid hyphae by fusion has formed a dikaryon. Dikaryon nuclei has been fused to form a zygote, right, which is called as synkaryon, and they will produce a fruiting body in which haploid spores are produced by meiosis. Meiosis is reductional division, and by that haploid spores are formed, and these spores on germination will produce again haploid hyphae, right? This is a simple cycle. Okay, it's also given in the NCERT, you can check there also. So I am covering everything in the NCERT plus your competition requirements. I hope you will like it. Next is the basis of classification. So we are classifying kingdom fungi into further classes, and these classes are formed on the basis of morphology of mycelium. Morphology of mycelium means how they have the mycelium. They are filamentous form, they are oval in form, or like they are septate or aseptate. These things we are looking here. Mode of spore formation, how they are forming the spores and their fruiting bodies. So all these things we are using to classify kingdom fungi. So let's see very first Phycomycetes. Why they are called as Phycomycetes? Because of their resemblance with algae. Like phyco word is usually used for the algae and myco for the fungi. And here we are saying Phycomycetes means they are called as algal fungi because of their resemblance with algae. They are not manufacturing their own food, but they are like the thallus in aquatic environment. That's why they are called as Phycomycetes. Okay. So let's find out where they are present. So they are present in different environments, like they can be found in the aquatic habitat, they can be found growing on the wood in damp places as obligate parasites. What are these obligate parasites? Obligate parasites are the organism which cannot complete their life cycle without specific host. So they have to parasitize a specific host to complete their life cycle. Okay. Next is the mycelium. So they are having aseptate and cynocytic mycelium, or you can say hyphae. So aseptate means they are not having septa, so they will be multinucleated in structure, right? Another thing, how they are reproducing asexually by zoospores and aplanospores. So I already told you in aquatic environment they produces zoospores, that are the motile spores having flagella, right? Another aplanospores, these are non-motile and released in the air, right? Okay. Spores are produced endogenously in sporangium. So how spores are produced? Spores are produced in sporangium. Like the Rhizopus we have studied, there is the bulb-like structure called sporangium which splits to release the spores outside, and that spores germinate, right? What is zygospore? In sexual reproduction, this zygospore is formed by the fusion of two gametes. Okay, so two gametes means we can say like male and female, or we can say like two different strain gametes have been fused to form the zygospore. Like here is given, gametes can be a similar or dissimilar. Similar gametes means they are exactly similar morphologically. When you see their sizes, they are exactly similar. So they are called as isogametes. And dissimilar means they will not be same size. Like one will be smaller size and other one will be of larger size. Or we can say like one is motile, like smaller motile male gamete, and another one is larger non-motile. So they can be like that also. Clear? So there are different forms of gametes: isogametes, anisogamous, or oogamous, right? So example comes in mucor, rhizopus, albugo, and an important thing here is given Rhizopus, which grows on the bread and called as bread mold. Albugo candida, it is a parasitic fungi. Remember this thing. Very important. Albugo candida is a parasitic fungi on mustard leaves. Okay, and you will see that on mustard leaves there are white spots present. These white spots are because of albugo. Okay, remember this thing. So here I am having life cycle of Rhizopus. How Rhizopus is growing. So there are two strains. So we are signifying different strains by plus and minus simply just to signify that they are genetically dissimilar. Okay, so these two strains hyphae is mating. So this is called as plasmogamy, right? So they are getting fused together, and what they are producing, zygosporangium, or you can say like zygospore. It is heterokaryotic. Heterokaryotic means hetero means of different, karyote means nucleus. So it is having two different strain nucleus, right? That's why it is called as heterokaryotic. So these zygosporangium on germination produces a new hyphae, and in this one of the specialized hyphae will produce a sporangium. This sporangium is producing spores inside it by a process meiosis. So by meiosis haploid spores are formed. These haploid spores have produced haploid hyphae. These haploid hyphae have combined together to form diploid zygospore. Simple, and that germinated to produce again haploid spores by meiosis. Clear? This is the general cycle of the Rhizopus. You can remember this, right? Okay, great. So here actual picture of the stained Rhizopus is shown under the microscope. Clear? Okay, let's move to the next life cycle. So here is the another life cycle in which asexual and sexual phases are shown, and there is somewhat different mode of reproduction. So first of all, let's look at the asexual mode. So here you can see a specialized hyphae has formed zoosporangia, and they have produced motile zoospores. Right, you can see here two flagella are present. These motile spores can use their flagella to move in the water body, right? Sometimes they lose their flagella and get encysted to protect themselves from unfavorable conditions. Like, so they can again get motility, they produce candy juice spore, and they can also lose this flagella, okay, to form encysted secondary juice spore. So these juice spore on germination again produces mycelium. Simple is that, okay, asexual reproduction specialized zoosporangia has been formed. That zoosporangia on in them have produced zoospores that are flagellated zoospores. These zoospores on germination produced again a mycelium, right? Another is the sexual phase. In this you will see that One mycelium is specialized into oogonium and another hyphae has produced antheridium. This antheridium has covered the oogonium, right? And through fertilization tube, they have released the sperms. These sperms have entered one of the egg and fertilized it to form zygote. Okay, this zygote is now called as oospore. We have talked about it, oospore. And oospore on germination again give rise to mycelium. Right? Clear? So this is the general cycle. Clear? Next is the Ascomycetes. Ascomycetes. Why they are called so? Because they are producing spores in ascus, right? That's why their name is Ascomycetes. And they are called as sac fungi. Sac means hollow structures, like in this ascus is present in which spores are produced. That's why they are called as sac fungi. Like, clear? Okay. In Ascomycetes, so it includes multicellular as well as unicellular organisms. In the multicellular organisms, it includes like penicillium, and in unicellular, it includes like yeast. So simple it is, yeast comes in Ascomycetes. Okay, these are saprophytic, decomposers, parasitic, or coprophilous. So this new word coprophilous means growing on dung. So these grow on the dung. Okay, you have to remember NCRT covers this thing, coprophilous. Clear? How is the mycelium? Mycelium is branched and septate. Septate means having septa between the nuclei. Right? Okay. Asexual spores are conidia. So they are producing conidia exogenously. Remember this thing, exogenously in conidiophores. So you have observed this thing that they have produced conidia exogenously. Okay, let's check out the picture. Here you can see. So they are produced outside the main body. So that's why they are called as exogenously. Right? Same in the penicillium and aspergillus. They are produced outside the body. Right? So I already told you endogenously means inside the body and exogenously means outside. Right? Sexual spores. Sexual spores are ascospores, right, which are produced endogenously in sac-like asci. So here comes an important point. Asexual spores are conidia produced exogenously, while sexual spores are produced endogenously, means inside the body. Correct? Okay. Minimum 4 ascospores are formed in one ascus. In one ascus, minimum 4 ascospores will be produced. Generally, there are 8 ascospores. Clear? In one ascus. Okay, so these are the ascus, and in this you can see these ascospores. Clear? Okay. The asci are arranged in fruiting bodies called ascocarps. So here you can see this is ascocarp in which these ascus are present. Clear? Okay. Example, Aspergillus, Penicillium, Claviceps, Neurospora. These all comes in this Ascomycetes. Right? What are the economic importance, and from here questions usually comes. So you have to remember Neurospora. Neurospora is used in biochemical and genetic work. So usually it is asked which fungi is used extensively in genetic work. So very simple answer is Neurospora, right? Next is the morels and truffles. So these are edible and are considered delicacy. Okay, they are eaten. Saccharomyces, we have already studied about the Saccharomyces cerevisiae. So this is fermenting agent, right? This is used in the bakery for the production of bread and beverages also, right? So because they are used in bakery, they are called as baker's yeast, and because they are used in brewery industry, they are called as brewer's yeast, right? So you can remember this thing: baker's yeast, brewer's yeast. Clear? Okay. Penicillium as a source of antibiotic. Penicillin. You already know that. First antibiotic produced was penicillin by Alexander Fleming, right? You remember that? Clear? Great. So here is the life cycle. You can see here these ascospores on germination producing this mycelial body. Like, clear, these are the haploid because haploid ascospores has produced this. So suppose there are two different mating types, plus and minus, we can represent it like, right? So first of all these will fuse together, and this fusion is called as plasmogamy, right? After plasmogamy they have shifted to dikaryon stage. So here it comes the dikaryon stage, which was absent in Phycomycetes. So here the dikaryon phase having two nuclei. Now they have produced ascocarp. This is the fruiting body having dikaryotic asci. Dikaryotic. Remember this thing, having two nuclei in the ascus. Clear? Now they have undergone meiosis to produce ascospores. So they have produced the ascospores. This is the real picture, right? You can observe clearly ascospores here. Clear? Again these ascospores will germinate to produce hyphae. Clear? I hope you remember that, and it's easier to learn through the cycle, right? Okay. Next is Basidiomycetes, or it is called as club fungi. So it is a common group which includes mushrooms, bracket fungi or puffballs, right? They grow on soil, on logs, tree stumps as parasites, example rust and smuts. So in this group the most important is these rust and smuts. We will talk about it later on. Mycelium. How is this mycelium? Branched and septate. You have to remember this. Mycelium is branched and have septa. Asexual spores are not found. Remember, asexual spores are not found in Basidiomycetes. Another thing, vegetative reproduction is by fragmentation. Simple fragments can grow into a new individual, right? Sexual reproduction. Sex organs are absent. There are no sex organ. Plasmogamy by fusion of two vegetative or somatic cells of different strains. So here we can also write like plus and minus strains that fuse together, and the fusion of that hyphae is called as plasmogamy, right? And then karyogamy, fusion of nuclei, right? These nuclei fuse together to form a zygote, and meiosis occurs in that. So here meiosis occurs in Basidium, which produces four basidiospores, right? And these are produced exogenously. Remember this thing, exogenously they are producing it. Okay, example are agaricus, mushroom, Eustilago, smut, Puccinia, rust. Clear? Here is the life cycle. So here you can see we have taken the different strain minus and plus. They are fusing together to form a dikaryon. Here the dikaryon having the two nuclei, they are present at the lower side in the gills. So these are the basidiocarp, fruiting body, right? In this gills, you must have seen at the below part of the mushroom there are gills. In that gills, basidia are present. These basidia undergo karyogamy, means fusion of nuclei. Later on, undergo meiosis to form basidiospores, outward, right? Exogenously. Clear? These basidiospores germinate to produce haploid mycelium and the cycle repeats. Simple, right? Here is the microscopic structure has been shown to you so that you can easily identify this thing. Clear? Okay. What are the economic importance? Puccinia graminis causes rust disease. Why this is called as rust disease? Because it appears like a rust. Clear? So you must have seen the rusting on the iron. So it appears like that. Okay. It completes life cycle in two hosts, wheat and barberry. Clear? So it needs two hosts, wheat and barberry. Rustilago causes smut disease. Why these are called as smut? Because they mainly infect the seed and form black sooty spores inside seed. So inside seed they are producing black sooty spores. That's why they are called as smut. Clear? So they are mainly affecting wheat, maize, barley grains. Clear? Toadstool is a poisonous mushroom. So you must have heard about the mushrooms. Some are edible, some are non-edible, some are poisonous, non-poisonous. So toadstool is a poisonous mushroom. Okay. Agaricus mushroom, simple. You call like which are eaten, which are edible. Puffballs, clear, they are also edible. Clear. Next is the Deuteromycetes group. I hope you are getting my points easily. Are you getting? Okay. Deuteromycetes. These are commonly called imperfect fungi. Why they are called as imperfect fungi? Simple. Because only asexual phases are known. Sexual phases are not known. So if we identify some sexual phases in this group of fungi, so we shift it to the another groups. Okay, so we are covering here only imperfect fungi in this group, Deuteromycetes. Asexual spores are conidia, mycelium is septate and branched. These are also saprophyte parasite decomposers. So decomposers help in mineral cycling, you already know that, right? Okay, Alternaria, Colatotrichum, Trichoderma, these are all examples of Deuteromycetes. You can remember it like ACT, 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 Alternaria, Colatotrichum, Trichoderma. Clear? And here are the important points. Economic importance. So economic importance generally includes, we say like what is the importance, what are the positive impacts, but they also include their negative impacts. So remember, whenever you are looking for economic importance, look for the both sides, positives and negatives. So here Alternaria solani causes early blight of potato. You have to remember this thing. Colatotrichum fulcatum causes red rot of sugarcane. You must have seen sugarcane having red rot. Trichoderma, source of immunosuppressive drugs, cyclosporin A. Cyclosporin A, immunosuppressive drug. Remember, you have to keep it in mind. It may come in the exam also. Okay friends, have a nice day. This is all for today. I hope you like this video and you have cleared all your doubts and concepts. Okay friends, have a great day.